Hello, this is a video lecture for Microbiology, Bio 2310 for, Tuesday, December 5th. And this is covering Chapter 14 in the book, Host Defenses, Overview, and Nonspecific Defenses. Our objectives of this chapter are to look at the defense mechanisms of the host, define the three different lines of defense, and to summarize the systems involved in immune defenses. And in this first chapter, we're going to focus on the second line of defense, nonspecific immunity. Okay, so if we look at the three different lines of defense, you can see here we have host defenses, and there are two basic types of defenses. First is the innate nonspecific defense, right here. And then the second is the acquired specific line of defense. Now, the innate nonspecific line of defense is split into two different lines. The first line of defense and the second line of defense. The first line of defense specifically incorporates physical barriers, chemical barriers, and genetic components. It has nothing to do with immune cells, and it basically is the anatomical protection of the body against foreign invasion. The second line of defense is a cellular response involving immune cells, including B cells and T cells that, I'm sorry, they don't include B cells and T cells, but this is very nonspecific. It includes the inflammation that occurs associated with pathogens, interferons, phagocytosis, and a whole cascade called complement. The acquired specific line of defense is the third line of defense, and it is naturally acquired through infection or maternal antibodies, and it can be artificially acquired through vaccination or some type of immune serum that contains IgG. Okay, so the first line of defense includes anatomical and physical barriers, and this keeps microbes from penetrating a sterile body compartment at the first place. The second line of defense is an internalized system of protective cells and fluids, and it includes inflammation and phagocytosis. And the third is a highly specific line of defense. It's acquired, and it is based on specialized blood cells called B cells and T cells. Okay, so looking at the first line of defense, one of the important portions of the first line of defense is physical and anatomical barriers to the body, and that includes skin. Okay, so the outermost layer of skin, called the stratum corneum, is composed of compact, tightly knit epithelial cells impregnated with an insoluble protein called keratin. And the outer layers of skin are constantly sloughing off. Their skin scales then carry microbes away from the body. And also hair follicles that are involved in the skin, hair is falling out, and as hair falls out, then that carries with it pathogenic microbes that could be invading the body, and this is called desquamation. And then sweat glands are creating an inappropriate environment, a harsh environment that is antimicrobial, and this is used to flush away microbes. Okay. Also, another barrier is mucous membranes. And mucous membranes are lining the endothelium of much of the tissues in the digestive tract, the urinary tract, respiratory tract, and also in the eye. A coat of mucus will, on some of these membranes, will impede the attachment of bacteria. The mucus on the membrane actually flushes away bacteria before it can adhere to the endothelium. The eye is coated with mucus, and attack is prevented by blinking and tearing. And saliva also carries away microbes into the harsh environment of the stomach. And then just releasing microbes from the body, when the body is invaded by microbes, then it has the ability to vomit which is also called emesis. You'll hear emesis a lot in a hospital environment, and then also by defecation. Okay. Mucous membranes also line the respiratory tract, and your nose is lined with nasal hair, including larger hairs or smaller fine hairs that are called cilia. And then the respiratory tract is lined with mucus and other fluids associated with infection. When the respiratory tract experiences infection, then the fluid production increases to rinse the fluids out of the respiratory tract by coughing and sneezing, runny nose, things like that. And then the respiratory tree is ciliated. That includes the bronchial tubes and the alveoli. The epithelium in the respiratory tract is ciliated, meaning that there are very, very many small hairs where potential pathogens can be caught and then forced back out of the lungs. Okay, and then genital urinary tract. There's a continuous urine flow through the ureter and through periodic bladder flushing, and urine flow keeps the genital urinary tract sterile. And then vaginal secretions, which provide a harsher environment for foreign invaders like bacteria. Then those secretions are flushing the foreign invaders outside the body. Okay, so here's just a overview of the first line of defense. You've got saliva. Saliva includes antibacterial enzymes like lysozyme, which works directly against peptidoglycan. Tears also have antibacterial enzymes, including a different type of lysozyme. Mucus will trap dirt and microbes and flush them away. Skin prevents entry into the body. Stomach acid at low pH will kill harmful microbes. And then good gut bacteria will outcompete and bind up all the receptor sites over and against bad bacteria, and prohibit the bad bacteria to actually bind cells. And going to good bacteria, we call this your resident microbiota, and this blocks the surface of the epithelium of different tissues and mucous membranes, and prevents pathogens from passing and getting access to cells. The resident microbiota actually form a biofilm, and the biofilm prohibits the pathogens from coming in. And it also can create an unfavorable environment. The good bacteria can create toxins that will wipe out the bad bacteria. Also, differences in pH, differences in secretions will create an unfavorable environment for the pathogens. And then commensal biota in the gut train the host defenses to keep commensals in check. So you do have some type of response, immune response against commensals, just so they won't overgrow. And also the commensals will train the host defenses to eliminate pathogens. And one of the problems where pathogens really start to crop up is when antibiotics are used, and antibiotics can interrupt this particular process by wiping out the commensals. The immune system doesn't know how to respond. The pathogens then take hold in the gut. This would be like Clostridia difficile, C. diff, and then you get an infection. There are also non-specific chemical defenses. Remember, in the first line of defense, we have physical barriers, chemical barriers, and then genetic components. And this is a part of the chemical barriers. This includes antimicrobial secretions from sebaceous glands and meibomian glands in the eyelids. Includes lysozyme. This is an enzyme, like I said before, in the tears and the saliva that can dissolve peptidoglycan cell walls. Sweat has a high lactic acid concentration that is toxic to cells, and then a high salt concentration that's also toxic to most bacteria except for things like Staphylococcus. Skin has a high pH, or an acidic pH, low pH, and a high fatty acid content, and these fatty acids have an antimicrobial characteristic to them. Stomach contains hydrochloric acid. That's the acid that makes the pH in the stomach so low. That also kills microbes. The intestines include digestive juices and bile. And then antimicrobials are present in the semen, and this will prevent the spread of infectious diseases through the urogenital tract. And then the acidic pH in the vagina keeps yeast overgrowth in check. Okay. Now, including the three types of defenses in the first line of defense are genetic differences in susceptibility. And so the body has genetic immunity, and all of us share a specific type of genetic immunity where this particular species, the human species, is immune to diseases of other hosts. We can't get specific diseases that animals get. There's very, very little transmission of things like bird flu and swine flu to humans. It can happen, but we do possess a genetic immunity, and these diseases need to mutate before they can actually invade a human host. There are still zoonotic infections that will attack a broad spectrum of animals like anthrax, rabies, H1N1 flu, where genetic immunity does not come into play because the host species, human species, is not immune to those diseases that are presenting in other animals. Okay, but there are interspecies genetic immunity differences. There are genes that code for the second and third line defense, and those then protect the body. So humans are not getting animal diseases. Humans don't get distemper from dogs. They don't get feline leukemia from cats because genetically we're different. And this is all outside of that last bullet. This is all a part of the first line of defense. So remember, for the test, the three types of immunity that you get are the three components of the first line of defense are physical barriers, chemical components, and genetic immunity. So remember that as the part of the first line of defense. Okay, so going back to the three lines of host defenses, we have the first line of defense, which consists of physical barriers, chemical components, and genetic immunity. Then we have the second line of defense, which is cellular immunity that is innate. Then we have the third line of defense, which is cellular immunity, which is acquired, and this is acquired through B cells and T cells. So those are the three lines of defense. The three components of the first line of defense, as I stated before, that's physical barriers, chemical components, and genetic immunity. And for different physical barriers, there are many different physical barriers that serve as defenses. Your skin, your eyelids by blinking, your mucous membranes in the epithelium, the harsh environment of the stomach. These are all physical barriers that serve as defenses. Now, looking briefly at the second and third lines of defense, this is the study of immunology, and these are the features of the body's second and third lines of defense. Then you have immune systems that are constantly roving the body doing surveillance. They recognize foreign materials, and they know the difference between good and bad foreign materials. You've got good foreign materials that are commensals, and then you have bad foreign materials that are pathogens, and they will destroy the pathogens and they will keep the commensals in check. Okay, and then you have white blood cells that are trained to differentiate between self and non-self. So the white blood cells will not attack the body, it will not attack self, but it will attack those pathogenic microbes that are non-self. Okay, and so when you look at white blood cells, here's a monocyte, and it is looking for specific cell surface receptors, and when the receptors bind between the white blood cell and normal self molecules, or normal self cells, then there's no reaction. Okay, it recognizes it itself, the white blood cell moves on. However, in contact with foreign cells, these white blood cells then will differentiate into macrophages, and the macrophages will recognize a pathogen here. The pathogen has specific molecules on the site called PAMPs, and I'll tell you what PAMPs are in a minute. And then the white blood cell phagocytizes the pathogen, the pathogenic microbe, eats it up, destroys it, and then eliminates the cell particles. Okay, and a pathogen, PAMP is a pathogen-associated molecular pattern. Okay, those are just molecular patterns that the body has already previously trained. This is in the second line of defense, not the first, but the body has been previously genetically programmed to see these particular molecular patterns and recognize these molecular patterns as foreign. Okay, so all cells have markers on them, and these markers are molecules on the cell surface used to identify whether the newly discovered cell poses a threat. Okay, and markers are usually made of proteins and or sugars. They can be glycoproteins, regular proteins, or they can just be sugars by themselves. And when a phagocytic cell sees a self cell, sees a non-foreign cell, leaves it alone. So when it sees a non-self cell, in specific instances, not all instances, it will destroy the cell by phagocytosis. And then there is middle ground where non-self proteins attached to cells are not harmful. When the body sees non-self proteins on these non-harmful cells, then it will leave those cells alone. However, with some commensals, it will keep them in check, so it will destroy a portion of them, but won't destroy all of them. Okay, and the immune system can go wrong. Okay, things can go wrong with the immune system, and this one is when you see allergies and autoimmunity. Okay, there's a person sneezing, they're allergic to cotton, you see the cotton balls in the background. You can get allergic reactions, including alopecia. The circular alopecia is associated with allergies. It can also be associated with stress. When you see the circular pattern of hair loss, sometimes this will happen due to stress. You can see eczema, which is an autoimmune reaction that happens due to overreaction of IgG cells to skin or IgG components to skin. And then inflammation is a good thing in the short term. Inflammation will rid the body and rinse out the pathogen and cause immune components to come in to attack the pathogen. But overinflammation, when inflammation is prolonged, can cause these different disease states, including cancer, cardiovascular disease, and so forth. Okay. So when we look at the systems involved in immune defenses, we need to look where in the body we see these. Okay. And primarily, these immune cells are roving and doing surveillance in what's called the reticuloendothelial space. And this is an interest in the space outside the cells, excuse me. It's a massive network of connective tissues. Okay. And these connective tissues contain extracellular fluid. Okay, you can also see these systems involved in the bloodstream, in the lymphatic system. Remember, we have a blood system and a circulatory system, and we have a lymphatic circulatory system. And so this just shows you all these different spaces coming into play. So we have lymphatic capillaries here. It's a part of the lymphatic system. We have blood capillaries here. Okay, then we have cells and tissues. And then we have the reticuloendothelial space here outside of the cells. And then you have some extracellular fluid that's building up around the lymphatic cavities. Okay. Also, you see extracellular fluid building up around the blood capillaries. Okay. So regardless of which component is first exposed, then an immune reaction in any one of them will be communicated to all of the others at some type of microscopic level. So all of these systems are put on high alert as soon as a pathogen has been detected. Okay, so the reticuloendothelial system consists primarily of the thymus, and this is where important blood cells, white blood cells, mature. This is where you've got T cells. T stands for thymus. B stands for blood. So T cells are maturing in this particular region. Also, the reticuloendothelial system exists in the lymph nodes, tonsils, spleen, and lymphoid tissues. And then there are MALT tissues. These are mucosa-associated lymphoid tissues, and wherever you find mucous membranes, you also see lymphoid tissues. And then the reticuloendothelial system itself is called the res, just R-E-S, and it teems with macrophages that are roving the body, and they're just waiting for attacking foreign intruders. This is a non-specific type of immunity, so these are non-specific immune cells along the second line of defense. Then along with the reticuloendothelial system, you have the lymphatic system. And this was a network to transport lymph throughout the body. Provides an auxiliary route for the return of extracellular fluids to the circulatory system. So the circulatory system and the lymph system are attached. And this is a drain-off system. So when you have a large inflammatory response and inflammatory mediators, cells, and fluid are locked in tissues, then this particular response then allows these inflammatory mediators to be able to drain off. Okay, and the lymphatic system is home to white blood cells, and these are used for surveillance, recognition, and protection against foreign materials. Okay, and so when you look at the lymphatic system over here, then there are a lot of different portions of the lymphatic system. The lymphatic system is unidirectional, and the circulatory system over here is bidirectional. Okay, so there's bidirectional flow to and from the heart and the lungs, and there is attachment with the lymphatic system. Lymphatic system is shown here in sort of dark gray, and the lymphatic system is always from the extremities directly to the venous system. Okay, and there's crossover where there's lymph nodes along the venous system here. This shows the lymphatics in the mammary gland. Okay, so lymphatic fluid. This is formed when certain blood components move out of the blood vessels and migrate into lymphatic cavities. And lymph is primarily water, just like most of the body, salt, about 2 to 5% protein, where you see antibodies like the bloodstream, as well as albumin, again like the bloodstream. And these will transport white blood cells throughout the body. Okay. Lymphatic vessels mimic the lines of the blood vessels, and so they're constructed along those lines. Wherever you find the blood system, the blood circulatory system, you'll also find the lymphatic circulatory system. In fact, they've just discovered that the lymphatic system is also existing in the brain, and so lymph tissues are used to drain fluids off of the brain, and they mimic the flow of the circulatory system. And this is now not correct because it used to be thought that the nervous system did not have a lymphatic system. So this particular bullet is not correct because it says lymphatic capillaries are not in the nervous system. That is absolutely not true. However, you don't see the lymphatic capillaries in bone, placenta, or the thymus. Okay. And like I said before, the flow of lymph is unidirectional from the extremities to the heart. And you do have a lymph pulse. You can measure the lymph pulse. It is not the same as a circulatory pulse, but lymph is moved through contraction of skeletal muscles via lymphatic ducts, and this is involuntary response, and so the muscles contract, and that allows the lymph to move back to the heart. Okay. Now, in terms of the second and third line of defense, or primarily the third line of defense, we have the thymus. The thymus is the site of T cell maturation, and it is the largest in its existence. It is largest at birth, proportionate to the human body, and it exhibits the greatest activity during childhood through puberty. And thymocytes, which are stem cells in the thymus, develop specificity, and then they're released to become mature T cells. And then the T cells migrate to other lymphoid organs. Okay, so here's the thymus. The thymus is full of blood vessels. It also has a center portion called the medulla and the outer portion called the cortex. Okay. Other lymphoid organs include lymph nodes, which are scattered throughout your body. These are associated with large blood vessels in the thoracic and abdominal cavities, and they filter out materials that have entered into the lymph. And they provide appropriate cells and niches for immune reactions. So this is where immune reactions can take place, and this is where the cells can be released for immune reactions. Another lymphoid organ is the spleen. It serves as a filter of the blood, okay, instead of the lymph. The spleen removes old blood cells and filters pathogens from the blood. And then phagocytosis of pathogens occurs in the spleen by resident macrophages. Okay, and here's just a figure that shows lymph nodes scattered through the body, and it does mimic the circulatory system. Okay, here's a picture of a spleen. You can see it's quite large, and it's engorged with blood because it's filtering out blood. Okay, other lymphoid tissues include the tonsils, which provide an active source of lymphocytes. The breasts of pregnant and lactating women. This is where lymphoid tissue is producing antibodies, and the antibodies then are passed maternally to the child. And this is important for early immunity. As the immune system of the baby is developing, then breast milk is providing the antibodies, maternal antibodies, that help the baby to ward off foreign attacks. And then you have gut-associated lymphoid tissue. This is like malt, only it's galt. And you see this in the gut, the appendix, lacteals, which are not a part of the gut, but they are associated with galt, and also Peyer's patches, which are large cell bodies that are associated with gut lymphoid tissue. And galt provides primarily immune functions against intestinal pathogens. Okay, and there's also malt, salt, and balt. Let's see, malt is for mucosa associated, salt is for, I think that's serous membrane associated, and balt eludes me. Oh my goodness, I should know this, but balt, I think, is bronchial associated lymphoid tissue. Okay. The gut endothelium is lined with Peyer's patches. Here's an example of a Peyer's patch. This box is around a Peyer's patch in the ileum, and these Peyer's patches are producing secretory IgA that is secreted into the gut itself through the microvilli, and these secretory IgA molecules are roving the body and looking for foreign invaders. Okay. In terms of the bloodstream, this is the circulatory system, the heart, the arteries, veins, and capillaries. It's parallel and interconnected and complementary to the lymph system. And circulating through the bloodstream is whole blood. This is blood cells suspended in plasma. And then quite frequently, you'll hear the term serum. And serum is, not a physiological component. It's actually an artificial component that you see if you take a blood sample, you clot the cells, and then you retain the clear fluid. The clear fluid from clotted blood is called serum. Okay. And if we look at unclotted whole blood, we have the plasma. The plasma is the component that is the fluid without the cells. Okay, it contains lots of proteins like albumin. And then in whole blood, you've got the buffy coat, and the buffy coat, which sounds like Buffy the Vampire Slayer, um, contains white blood cells, and then the bottom portion, which is the hematocrit, contains red blood cells. Okay, if we clot the whole blood, then you have serum on top of a clot. The clot is the bottom portion, the serum is the top portion, and the proteins in the serum are different than the proteins in the plasma because you're losing a lot of the proteins associated with clotting in the serum. Okay, so plasma itself, that is the white or the non-cellular component of whole blood, is water. It contains proteins, contains globulins, including antibodies like IgG and IgM. It contains other immunochemicals, clotting factors, the entire clotting cascade. You'll find primarily in the plasma, hormones, nutrients, and dissolved gases. Also waste products that will be filtered out in the spleen and the liver and the kidneys. Okay, and then outside the plasma, you also have blood cells. And the production of blood cells we call hematopoiesis. This is production of blood cell occurs in the yolk sac and the embryo, the liver and lymphatic organs in the later embryo, and the red bone marrow in the fetus. Okay, and this is where you'd find stem cells. And these are primary precursors to new blood cells, and these are maintained in the marrow, and they continue to be produced in the marrow throughout the lifetime of the individual. Stem cells mature and differentiate. They become other things. They go from immature and unspecialized to mature and specialized. So the stem cells provide the components for the blood. Okay, and if we look at the different types of blood cells, there's red blood cells or erythrocytes. There's white blood cells, which are called leukocytes. And then there are very small cells that are non-nucleated called platelets, and these are called thrombocytes. Okay. White blood cells can be divided further into those that have granules, and these are granulocytes, and those that don't have granules, which are called agranulocytes. Okay. And if we look at the trajectory of white blood cells, you can see a trajectory of a lot of different blood cells. You can see a hematopoietic stem cell here in the bone marrow. The stem cell will differentiate into another stem cell for blood cells except lymphocytes. Okay, lymphocytes are a specialized type of white blood cell. The hematopoietic stem cell can go any other direction and make a lymphoid stem cell. The lymphoid stem cell becomes lymphoblasts on their way to becoming mature agranulocytes, T and B cells. Okay, agranulocytes, T and B cells provide immunity through the third line of defense. That's specific immunity. Okay, and they are called agranulocytes because they don't have granules. Okay, and then there's another type of lymphoid stem cell called a natural killer cell. It's related to a T cell, but these cells are very, very active natural killer cells. You'll see in cells that are infected with viruses, as well as the cells that are cancerous, will attack these cells. Okay. And then stem cells for blood can go to a trajectory where they become an erythroblast, and then finally an erythrocyte, a red blood cell. They can become a megakaryoblast on their way to becoming a platelet, or they can become a lyeloblast to make granulocytes. These are neutrophils, basophils, and eosinophils. And another granulocyte that's mainly you see an allergic response. These are called mast cells. Okay, and then finally the stem cell can become a monoblast. Monoblasts become monocytes. Where's my arrow? Monocytes. Monocytes then can differentiate into macrophages or dendritic cells, and macrophages and dendritic cells are examples of phagocytic cells that can uptake components to undergo phagocytosis. Okay, so that gives you an overview of the different types of cells. I'm not so concerned that you know the different types of stem cells and different types of undifferentiated cells, but these differentiated versions, you should know T and B cells, you should know monocytes, macrophages, and dendritic cells, natural killer cells, eosinophils, basophils, neutrophils, mast cells, red blood cells, and platelets. You should know all these different end cell types and how they function in the body. Okay, so cells that have granules we call granulocytes. That includes neutrophils, that it makes up the vast majority of all leukocytes, all white blood cells. These produce toxic chemicals, and they're also phagocytics. They can phagocytose in the early stages of the new immune response. They are nonspecific in terms of phagocytosis. Then you have eosinophils, and these eosinophils have granules that contain oxidase, which will oxidize tissues, lysozyme, other digestive enzymes, toxic proteins, and inflammatory chemicals. So they will attack and destroy large eukaryotic pathogens, especially helminths. They're also involved in the formation of fetal tissues, and you do see eosinophils in inflammation and allergic reactions. Okay, then you have basophils, which are the scarcest type of leukocytes, and they are very much parallel to eosinophils, and they contain potent chemical mediators for digesting the bad guys. Okay. Then agranulocytes include lymphocytes, and lymphocytes are the second most common type of white blood cell in the body, and the first being neutrophils. And there are B lymphocytes, and these mature in special bone marrow sites in humans. And then there are T lymphocytes that mature in the thymus of all birds and mammals. Okay, and both populations of cells, T cells and B cells, are transported through blood and lymph. And these are the key cells in the third line of defense. This provides the specific immune response. Okay, and here are some white blood cells, some leukocytes surrounded by erythrocytes. This is a phagocytic cell. It's a large macrophage. Okay, and if we want to take a deeper dive into B cells and T cells, B cells contribute to antibody-mediated immunity. They're activated by foreign antigens, and they divide and they form specialized types of cells called plasma cells, which each produce a different type of antibody. And antibodies are just large protein molecules that will bind selectively to the antigen and participate in the destruction of the pathogen. Okay. T cells contribute to cell-mediated immunity, and they modulate immune functions, and they also kill foreign cells. So antibodies coming from B cells will tag the foreign cells, and then the T cells will participate in their destruction. Okay. Another type of agranulocyte is a monocyte. It's the largest of all white blood cells and the third most common in the circulatory system. And the cytoplasm has many vacuoles that has digestive enzymes, and these are discharged by the bone marrow. They go into the bloodstream where they exist as phagocytes for just a few days. And this is where monocytes become macrophages. And the macrophages are formed when the monocytes leave circulation and they undergo their final differentiation. So they leave the circulatory system, and then they differentiate as they enter the reticuloendothelial space. And these are responsible for many specific and non-specific phagocytic and killer functions. They will process foreign molecules and present them on the lymphocytes. This is called antigen presentation. And then they secrete biologically active immune molecules that are participating in the immune response. Okay, here's a monocyte before it's differentiated into a macrophage. Here's a macrophage killing a foreign invader. Okay. Then we have erythrocytes and platelets. Okay, and at this point, I am going to stop this particular video. There will be two videos for this particular chapter. This is the first video. It lasts approximately 44 minutes, and there will be a second video on YouTube. You need to be able to watch both of them.